Remember we said all cells of an organism have essentially identical genomes? Different cell types, remember, come from differential gene expression. Well, so what if there are abnormalities? Many times this will lead to cancer. Regulation of gene expression usually occurs by regulating transcription. Now, it might be helpful at this point for you to study slides 25 through 30. And if you haven't gone to YouTube and watched the protein synthesis lab review that I posted, you might want to do that if you have any questions about protein synthesis. It's interesting because differential structure of chromatin also helps regulate gene expression. Genes with heterochromatin don't get expressed. We can also chemically modify histones, remember those are the proteins bound to the DNA, and DNA to vary structure and gene expression. For example, if we talk about histone acetylation, acetyl groups attach to the amino acids in the histone tail. This opens up the chromatin structure and actually promotes the start of transcription. Methylation is different. If you add methyl groups to the chromatin, you actually condense the chromatin and you make transcription more difficult. It actually decreases the rate of transcription. Remember we talked about genomic imprinting? Well, methylation is going to regulate which alleles. Does it come from the mom or the dad? Which of those alleles actually get expressed? These chromatin modifications do not, and this is very important, they don't change the DNA sequence. But they still can be passed to future generations. And this is a phenomenon called epigenetic inheritance, where it is the inheritance of traits that doesn't involve the nucleotide sequence. It's quite fascinating. So all of this acetylation, methylation, that type of thing can be passed to the children. Chromatin modifying enzymes initially control gene expression by increasing or decreasing DNA binding to transcription machinery. Most eukaryotic genes have multiple control elements. Now, control elements are non-coding DNA segments, which means they're not coding for a protein, but they can bind transcription factors and help regulate transcription. And this is going to allow eukaryotic genes to be controlled in their expression to a very, very precise degree. General transcription factors are essential for the transcription of all the genes coding for proteins. In other words, you are not going to transcribe those genes without the presence of the general transcription factors. Everybody has to have them. High levels of transcription require what we call control elements that interact with specific transcription factors. Now, let's talk a little bit more about the general transcription factors at the promoter. They are required by RNA polymerase to start transcription. It won't even start if they're not present. It is a must for every gene that codes for a protein. Some of them bind to the TATA box in the promoter. Some others will bind to other proteins, like example, maybe other transcription factors we'll be talking about, or maybe RNA polymerase 2. RNA polymerase cannot move on the template DNA until you have a complete initiation complex. And then it will produce that complementary strand of RNA. If a gene is not normally expressed, high levels of transcription are going to require an additional assistant. These will be the specific transcription factors. Those called proximal control elements are close to the promoter. Distal control elements, also called enhancers, are not close to the gene, or maybe they're in an intron. And the enhancer, remember, these distal control elements are going to be associated with only one gene. So it can become terribly complicated with the number of factors that are involved in getting genes expressed. Now, activators are proteins that bind the enhancer to increase gene transcription. They are proteins, right? And they have two domains. One of those domains is going to be used to bind DNA. One of them actually activates transcription. And when they are bound, this increase in protein-protein interactions causes gene transcription. Bound activators contact what we call mediator proteins, and they interact with general transcription factors, and they're responsible for making sure that the pre-initiation complex is put together correctly and is located in the right place. Study the slides in the PowerPoint for this section, and I think it will help you to visualize this. Some transcription factors, called repressors, inhibit gene expression. Some activators and repressors are going to increase and decrease, respectively, transcription by actually changing the chromatin structure. Control elements activate transcription only when the correct activator proteins are present. There are things called coordinately controlled genes in eukaryotes. These are genes that are typically co-expressed, but they're not organized into operons. These genes are scattered over different chromosomes, but they have the same combination of control elements. Activator proteins in the nucleus can recognize these specific control elements and promote simultaneous transcription of the genes, even though they may not be near one another or even on the same chromosome. Chromatin loops from different chromosomes may come together with transcription factors and RNA polymerases, and we think that these are probably areas that have a common function, that are specialized. Take a look at slide 55. We also can regulate gene expression in a post-transcriptional way. And this is very important because it allows cells to fine-tune their gene expression, particularly if the environment is changing. If the environment is changing rapidly, the cell needs to be able to respond rapidly. One of those is what we call alternative RNA splicing. So important. So this means that you can actually produce different messenger RNAs from the same primary transcript by just, if it changes, what are the introns, what are the exons, then you won't get the same messenger RNA. And this expands the potential of the genome tremendously. And scientists think this is probably a good explanation as to why humans have such low number of genes, because more than 90% of genes experience this alternative RNA splicing. Translation initiation of those messenger RNAs can be blocked by binding of regulatory proteins. Also, translation of all messenger RNAs in a cell may be simultaneously regulated. So, for example, we have what are called translation initiation factors. These are simultaneously activated in the egg after fertilization so that all of the proteins that are necessary once the egg has been fertilized get activated simultaneously. Eukaryotes typically have much longer lived messenger RNAs than prokaryotes. In eukaryotes, nucleotide sequence that regulate lifespan of the messenger RNA is located in the untranslated region at the 3' end. And looking at that term, one of the things that I find very confusing in the textbook is the way that they continue to abbreviate things like untranslated region, UTR, because by the time I have read a few more paragraphs, I can't even remember what UTR is. So I would encourage you, as you're taking notes, go ahead and spell these things out and begin to accumulate lists of who are the players, what are their jobs, what do they do, how do they function.